Badrinarayanan, who joins us from NCVS Bangalore. Anjana is going to be talking about searching for homology in vivo dynamics of bacterial homologous recombination. And Anjana also tells me you can interrupt her during her talk if you have any questions. Just put up your hand and one of us will bring up a mic to you. All right, take it away.
Thanks, Vaishnavi. I usually start by making a joke about how my name got pronounced. First time, I don't get to make that joke. Thank you for that. And thank you also for the invitation to be here. It's really an absolute pleasure. I want to say I might be the outlier for today's session at least. I am a biologist, and there are no equations in today's talk. It's going to be a lot of experiments, movies, and hopefully a lot of discussion that comes from it. Let's see if I can get this to work. Can you see anything? You see it? Oh, there you go, you see it. Okay, excellent. Okay, so I'm going to tell you about a story that we've been working on in my lab for the past 6 years or so, but it originated even earlier when I was doing my postdoc in Michael Laub's lab, and it is to try and understand the mechanism of homologous recombination, which is a very fundamental process across domains of life. And the example I'm going to tell you about today is the example of a bacteria. This little rendition you see is DALI imagining what DNA damage looks like. It looks quite futuristic, I would like to say. But if I didn't say it was DNA. So I was told if I do this, then the recording people who are going to watch the recording can see the pointer. In case it all fails, I'll come straight to you. Thank you. All right, so why should we think about DNA damage? Now, fundamental to the propagation of life is the maintenance of DNA. Francois Jacob said that the dream of every cell is to become two. And for that to happen efficiently, DNA needs to be replicated, duplicated efficiently. And we know a lot about this process, and I think we will have a talk later in this conference talking about the dynamics of the replosome. But you know, this molecule that's so essential for life is also highly unstable. The minute metabolism starts in our cells, cells are going to face DNA damage. This comes from intrinsic agents, but it also comes from extrinsic agents such as UV radiation, etc., which can do all sorts of damage to DNA, modify the chemical bases, but also cause breaks, physical breaks to the DNA, single and double-strand breaks being one of the most lethal forms of DNA damage. And so we now know that the maintenance of life not just requires the faithful duplication of DNA, it also needs the preservation of genomic integrity. And that's what we study in my lab. We try to understand how cells manage DNA damage and fix the DNA damage in a manner that allows for error-free correction, but still has some space for error, which allows for mutagenesis and evolution over time. So together, the ability to repair damage efficiently allows cells to toggle between genome maintenance on one side and genome evolution on the other side. And to ask most of our questions in the lab, we use bacteria, and this is the model system. I want to orient you guys to this very, very quickly because it's not E. coli. And very little in bacterial biology is E. coli. So this is Colobacter crescentus. It's an alpha proteobacter which has a dimorphic life cycle. It starts as a single chromosome cell called the swarmer cell. And then it differentiates into a stalked cell where it starts the process of replication. Now, once it starts replication, okay, now too bad for the recording people. I'm going to go and do it this way. So once it starts replication, so you see this at the inner circle, the DNA. The outer circle is just the cell. It completes duplicating its genome, and then it divides asymmetrically to give you two cell types. A swarmer cell again that needs to differentiate before it starts replication again, and a stalk cell that can replicate already immediately. And it'll become quite evident to you as I proceed in the talk as to why this is a really powerful system to study the processes I'm going to tell you about today. I'll also thank already the people in the beginning. All of the experimental work I'm going to tell you about was carried out by Afroz Chimtanawala in my lab, a graduate student who's now recently moved on to a postdoc at NYU. And some of the new movies I'm going to show you towards the end of the talk are done by a new graduate student in the lab, Saurabh Bhattacharya. And we also had an absolutely fantastic collaboration with Madan Rao, who's also in the audience. And I specifically want to thank also a postdoc in his lab, Jyotsna Palmer, who helped us with a lot of image analysis as well, and these guys for funding. All right, so the type of DNA damage I'm going to tell you about today is a double-strand break. Now, what is a double-strand break? Well, it's quite simple. It's when both strands of your DNA get broken. Now, this is one of the most lethal forms of DNA damage. If left unrepaired, it will eventually lead to cell death, but also underlies many pathologies, including cancer. Now, across domains of life, a faithful way of fixing a double-strand break is via a process called homologous recombination. Now, this process requires an uncut copy of duplex DNA that's used as a template for repair. And this process has been really very, very well studied, both biochemically and genetically since the 1960s, and we know what the essential steps of double-strand break repair are to allow for this template, to allow for this broken piece of DNA to use this template for repair. Now, I want to highlight two key steps here. One step is the process of end resection, where the double-stranded DNA is resected or chewed up to reveal these single-stranded DNA ends. Now, these single-stranded DNA ends are absolutely key because they are what's going to be used to do homology search, to find the right template match in the duplex DNA. And those single-stranded ends are then used as a strand invasion partner to allow for copying up whatever was lost at the break site. Now, this entire process, right, of finding the homologous partner and then doing strand invasion is carried out by one key protein. And in the talk, you'll have to remember only two proteins. This is one of them. This protein is called RecA in bacteria or Rad51 in eukaryotic systems. Now, RecA or Rad51 binds these single-stranded DNA ends once they are revealed and forms a nucleoprotein filament. And it's this filament then that does this process called homology search, to search inside the cell for the right base match. And the question that we don't have an answer to in the field, or are now beginning to get an answer to in the field, is the mechanism by which this RecA nucleoprotein filament actually goes about doing homology search. Now, what do I mean by homology search, and why is this a complicated problem? Now, it's easier to draw this out in this stick diagrammatic form, where your homologous template is right next to the break site. But inside the cell, the situation is really quite complex. I show you an example here of an E. coli cell with the break sites marked in red. Now, this break site, this double-strand break site, has to find its homologous template that's located here on the second chromosome. In an E. coli cell, this involves the sampling of 9 megabase pairs of DNA to find the one right match. And this process is 100% efficient. It occurs with high fidelity. So this break will rarely recombine with anything else on the chromosome. It's going to find that right match. And it has to do so in contention with a lot of ongoing processes in the cell. We know that the chromosome is highly compacted and organized. There's also other ongoing processes that are going to impede the process of search. So how a single break finds the homologous partner inside the cell is a rather complex problem. Mathematical modeling of such a search process, if it were to occur completely randomly, suggests that it can take anywhere between 3 days to a few years for one break to have found its partner. But inside the cell, we know this happens in a matter of a few minutes. So something is making this process rather efficient. Now, in the past, people have thought about this, yes?
How big are the breaks in terms of?
Okay, good, yeah. So, okay, excellent. So in vitro, Suri is asking how big is the break site, so how many bases are involved in the search process. So in vitro, it's been shown that 8 bases are enough to get efficient pairing. But in vivo estimates range from 1 kb to a few tens of kb involved in the search. But 8 bases are enough. Right, so people have thought about this search problem similar to how one would think about how a transcription factor finds its cognate promoter sequence inside the cell. You can consider various mechanisms. Once a transcription factor binds the DNA, it can go through 1D sliding, some sort of intersegment hopping, 3D diffusion, and then finally find its break site. In the case of a transcription factor, the problem is a little bit more simplified because you have n number of transcription factors looking for one promoter sequence. In case of the break that I'm talking about, it's one break looking for one sequence. So the target it's a bit more complicated in terms of finding your target. Stephen Kowalski's lab published a review which suggested a combination of hopping and intersegment hopping and 3D sliding to allow for the search process to occur efficiently. And this came actually from this very elegant experiment that showed that you need the ability of the chromosome to fold to bring two homologous strands closer to each other in close proximity. And so this is what the model looks like from a lot of in vitro studies to suggest that this RecA protein filament sort of binds to one segment of the chromosome and then goes through this hopping across these chromosomal regions, slides a bit also to finally come in contact with its partner. And so we can draw it out in this manner also. So there's the single-stranded DNA filament. It randomly collides with one segment of the chromosome, and then it does this sliding behavior as well as hopping across segments to finally land up at where its partner is. But if you take this now inside the nucleus or inside the cell, again, the spatial scale is a problem. So in case of a bacteria, one can imagine that this might still be okay. But in a nucleus, the nuclear volume is quite large for this break to go through the process I just told you about.
Anjana, sorry, I had a question.
Yes.
Is the sliding an active process?
Is the sliding an active process? No, so they do not think it involves ATP. In fact, RecA has ATP activity, but none of the search dynamic requires ATPase activity. So it seems to be a passive process of just hooking onto DNA and then transferring the filament onto a new segment. Yeah, so one of the ideas was that because in the nuclear volume this can become far more complicated, the idea was that search will occur in a proximity guided manner, in that you do all of these steps I told you, but you don't sample the entire nuclear volume, you only look around in a fixed area around your break site. This works out okay for eukaryotic cells where you have chromosome alignment soon after replication. But in bacterial cells, the chromosomes separate or segregate quite rapidly after replication. In fact, it turns out that even in yeast nuclei, where this has been studied recently, movement is a lot more than this proximity-guided search that people have previously proposed. Right, so, yes? What's the speed up you get? Yeah, so I will come to that in a little bit. It didn't seem to solve the problem. So the ELF lab, which had proposed how long it would take, came up with a much more simpler and elegant solution. And I'm going to just come to that in a moment.
Just a quick reminder, if you have a question, please put up your hand and I'll get the mic to you so that people online can hear you as well.
Right, I'll answer your question in the next slide. Right, so in any case, it doesn't seem like proximity-guided search is the solution to the problem. In fact, even this 1D sliding, 3D hopping alone can't solve the problem because it doesn't seem to speed the process up enough. So the ELF lab recently proposed a rather elegant solution to the problem. In their case, what they said was perhaps what happens is that this RecA nucleoprotein filament forms a stiff rod inside the cell, almost going end to end in a rod-shaped E. coli. And what then happens is that chromosomal regions come in contact with the stiff rod at random locations across the RecA nucleoprotein filament. And via that, what you can do is you can multiplex search. So instead of one RecA nucleoprotein filament searching one at a time, you have n number of searches coming from various parts of the chromosome colliding with the RecA nucleoprotein filament. And this sort of solved the problem quite a bit. They were able to build this model and show that if you do it this way, at least in E. coli, you can find your break site within half an hour, which was their estimate for homology search. Now, this model works okay, but it doesn't work when you take into account cellular geometries, for example. In bacteria alone, cells sample various types of geometries. Rod-shaped cells are just one variant of the many types of shapes you would get. And if you take the eukaryotic nucleus, for example, it's not a rod. So if you make a stiff rod, to get these collision events to occur at the frequency required seems to be less likely. There have been some studies in vivo looking at what this RecA nucleoprotein filament looks like, and it does form these really elegant filaments or bundles that span across the length of the cell. But again, what are the dynamics of this filament that allows for search to occur efficiently is not answered. And so this is the question I'm going to tell you about today. What did we find in Colobacter, trying to understand the search process? And so to do this experiment in Colobacter, we had to devise a system where we can site-specifically induce a double-strand break and then observe the dynamics of the break site. So we do this via engineering a double-strand break site. So it's called ISC1. It's a restriction enzyme site that we've engineered onto the Colobacter genome. The examples I'm going to show you, the break site is located at cell poles. So it's near the origin of replication shown here. Now, the site is recognized by an enzyme that cleaves the site to generate a double-strand break. And we regulate the expression of the enzyme in a manner that gets you one break only at a time. We do get some cells with two breaks, but then we do something to those cells. I'll tell you about it if you guys want to know. We also do make a genetic trick to isolate cells that have only one chromosome. So this allows us to look at the dynamics of search without repair. And then we do the same experiment in cells with two chromosomes to look at repair. And we remove replication from the mix, so the system is quite clean. We also mark the break site itself. So we add a protein that binds next to the break site. And this tells us where the break is, and it also tells us when a break occurs. And I'll tell you about how we do that. So here is the entire system. Is this visible or with the lights? Maybe we can dim the lights a bit because there'll be movies shortly. Okay, oh wait, there's this projector thing, right? Let's see. There was a projector. Okay, something's happening, I think. Anyway, I'll keep going, and then if we need, we'll call for help. Okay, thanks. Right, so I'll ask you to pay attention to the top panel first and then the bottom panel. The top panel tells you where the break is. So we start with cells with two markers on opposite cell poles. This tells us that both the chromosome sites are intact, nothing has happened to it. Then we express our enzyme that's going to make a double-strand break, and so you lose the marker at one pole, and this is because of this resection that I told you about. So you now have only one marker, and that's marking where the homologous template is. And at some point, you get the reappearance of the second marker. And this tells us that repair has been completed. So we can look at all the steps. We can see a break has occurred. We know where it is. And then we can see repair as well. In the bottom panel, I show you what RecA is doing in this movie. So we tag RecA with a fluorophore, and then we observe what happens to it. RecA has diffuse localization across the entire cell. When there is no DNA damage, no break. When a break occurs, RecA forms a localization at the break site. And then at some point, it engages in the process of homology search, looking for the template which is located at the opposite pole. And then once a template has been found, RecA dissociates and it has diffuse localization again. And RecA is not involved in the process of finishing repair. So it's really localized only when there's homology search. And so our estimates of homology search time also match quite nicely with E. coli's homology search time. It's approximately 36 minutes. And what I'm going to do for the rest of the talk is just focus on the dynamics at this time of homology search. And just to remind you again, what we did was to look at the RecA dynamics in cells with two chromosomes and then cells with one chromosome. One chromosome tells you about search, but no repair because there's no template. And then the two chromosomes where you can get repair also. And what Afroz did for the purposes of these experiments was to analyze two key properties of the RecA filament. She asked, where is the RecA filament with respect to the length of the entire cell? So for that, she maps the centroid of the RecA filament as it does homology search. And the second thing she asked was, what is the length of the filament and how does it grow and shrink during the search process? All right, so what does RecA do? So we localize RecA, and just to orient you, wherever you see the localization, that's where the break has occurred. And Afroz imaged RecA every 5 seconds, 10 seconds, 15 seconds, and 60 seconds. And so I'm going to play this out for you because it's so beautiful. Every 5 seconds, RecA forms these really beautiful helical structures, and then it takes off. And at every 60 seconds, it's out. It's gone from the break site and it's moving, goes all the way across the cell to the other opposite cell pole, turns around and goes back again. So as I told you, we're going to map the centroid position XC, an end of the music, but movie still goes on. Every 5 seconds, there's no displacement, so RecA is quite stable. And as you start to increase the time interval of imaging, you start to see this positive displacement. And what's really important is that there's no backtracking, so it's not moving back. It's mostly moving positively from where it was originally located. What's also really interesting is that once the filament reaches the opposite end of the cell, it turns around and it goes through this back and forth movement. And it does so several times before it finally finds its homologous partner. So I'm just going to show you this again. These are the chymographs where you have a RecA filament that's formed at the break site at one pole. And then you can track the movement across the cell, across time. And then you also get these back and forth movements. Yes, there's a question there. Maybe if you yell it out, I'll repeat it. I'll remember.
I don't quite understand the previous one, where if you're imaging every 50 seconds, shouldn't it be the same one as the 5-second one, but the 10th frame?
Every 5 seconds. So no. So this is every 5-second one, but the 10th frame. Yeah, so it does add up. So when you sort of collate the 5 seconds together, yep. Then you do get full displacement.
So it's basically every from 0 to 50, you're putting together all.
Yeah. So that does add up. So, but in 5-second intervals, there's no displacement. So every 5 seconds, there is no displacement.
But then why should the dynamics? Sorry.
No, go ahead.
But then why should the dynamics over 50 seconds or 1 minute be considered as like actual dynamics? It should be the 50-second one. The 5 seconds one should be the actual one, right?
Yeah, I think you and I are visualizing this differently. So when we do the imaging every 5 seconds, we're only asking what's the displacement between those two 5-second intervals. But what we are plotting here for the 30 seconds is when we've imaged at longer time scales. So there is displacement at longer time scales. You don't pick it up at the 5-second interval. That being said, if we were to image this for longer, where we would capture 30-second or 60-second displacement, then we'd pick it up. So this is only showing you for 1 minute, 5 seconds for 1 minute, for example. So we won't pick up as much positive displacement as what you'd see at these time intervals. Okay, so if we define what a traversal is, we define traversal by saying the movement of RecA from the pole where the break occurred to the opposite cell pole. It takes approximately 3 minutes or so for RecA to move from one pole to the opposite cell pole. And it takes about 6 to 8 such back and forth movements till RecA finally finds its homologous partner in the cell. So the question Afroz then asked was, is a homologous partner required for this directional motion? So in some ways, does RecA know that there is a partner in the cell? So this was an easy experiment for her to do. We could take these cells with just one chromosome and then ask what happens to the movement in this case. And in this case too, what we found was that RecA does move. So it starts at the break site, goes to the opposite cell pole, and goes back and forth several times. In fact, it didn't stop going back and forth for the entire course of our imaging because there was no partner to be found. It's very similar to what happens in life sometimes, an interminable search. Anyway, so how does RecA move, right? So as Vaishnavi was asking, is the sliding movement, for example, known to use ATP? We know RecA has an ATPase cycle. It can hydrolyze ADP anywhere across the filaments. When I say filament, you have to remember there are multiple RecA monomers sort of linked up with each other. And in the past, biochemical studies have suggested that RecA can go through ATPase activity anywhere within the filament. So it's not necessary that it would happen only at an end of the filament. So the idea that it could perhaps sort of treadmill on its own was not favored in the past because of this idea that you could have ATPase cycle happening anywhere. So what this suggested was that RecA filament can grow and shrink in its length, but it may not have enough motor activity to cause larger displacements inside the cell. And so the question then becomes, what's really driving this movement of RecA that we are seeing? In vitro, this has been observed quite nicely, the formation of a filament, some small growth and shrinkages in length, but none of this large-scale displacement. Okay, so we wondered whether there is a protein that drives it, and it's not RecA on its own. And so to cut a long story short, we arrived at a partner of RecA that's known to physically interact with RecA and is known to modulate various activities of RecA, including its ability to do strand invasion as well as homology pairing. And we know from our own work as well as work of others that RecN, which is a highly conserved protein across bacterial systems and also found in eukaryotic systems, is absolutely essential for double-strand break repair. And in our own system, you don't get any repair when we remove RecN from the mix. Okay, so what happens if you now remove RecN from the cells where we induce a site-specific break? And here, we notice something really stark. So in this case, RecA forms a filament at the break site, but it just doesn't move. And we can image this now for 100 minutes or so, and we don't see any movement. RecA is formed just fine at the break site, but we don't see any of this displacement that I showed you before. And just to remind you again, we know that without RecN, you do not get repair, which suggests to us that this movement that I just showed you is essential for the process of homologous recombination. And just to remind you again, we see that RecA remains rooted at the pole where the break occurred in the absence of RecN, but in the presence of RecN, it manages to traverse across the length of the cell in a matter of a few minutes. And this is true independent of whether cells have 2 chromosomes or 1 chromosome. In either case, there's just no movement without RecN. Very quickly, I'm going to summarize some key data. I'm not showing you any data for it, but I'm going to summarize the results. We found that RecN does not affect the rate at which RecA nucleates at the break site. So we measured this via looking at the intensity change of RecA once a break has formed, and the rates are similar. We also asked whether RecN affects the rate at which the filament grows once RecA has localized to the break site. And in this case too, RecN has no effect on the growth of the filament. And finally, we asked if RecN affects the filament stability of RecA at the break site. And here again, we find that RecN does not affect the stability of the RecA filament. So RecN is coming after the filament has formed. And we are able to show this also cell biologically, that RecN is recruited to the break site by RecA. And somehow after getting recruited to the break site, RecN is able to drive the movement of RecA inside the cell. And so that's the key question here. What's the mechanism that's driving this translocation? And I'm going to sort of end the talk in a few slides, but I am hoping that we can continue our discussion on what we think could be the mechanism of this movement. The one thing I do want to tell you a little bit about, though, is the fact that the translocation of RecA is associated with the remodeling of the RecA filament length. Now, what do I mean by remodeling? We see various shapes the RecA filament takes up during the process of movement inside the cell. It can be anywhere from a puncta to a long elongated filament, sometimes even a bent filament. Very rarely do we see 2 puncta, which may be marking the 2 ends of the break site, but we are able to observe that also. And in the absence of RecN, once RecA has formed a filament, we don't see so much length variation. While in the presence of RecN, you do get a lot of length variation once the RecA filament has formed. We're also able to show that this length variation is driven by the ATPase cycle of RecN. So RecN is part of a family of proteins that encompass the structural maintenance of chromosome class of proteins. These proteins are known to do various activities on the chromosome, including chromosome organization, compaction, loop extrusion, etc. And so what we can do is take these known mutants of SMC proteins that can block ATP hydrolysis or ATP binding and ask then what happens to the activity of RecN. And what we find is that when you block ATP binding, RecN does not affect the length of the filament. The length of the filament is long. But when you block ATP hydrolysis RecA forms these shorter filaments, which suggests that maybe RecN is using its ATPase cycle to modulate the RecA filament length, and that in turn that may be driving translocation of the RecA filament during homology search. All right, so what I've shown you today is that we can recapitulate the entire process of homologous recombination inside the cell, and we can perhaps now propose a new mechanism by which search occurs. And this search involves directional movement of RecA inside the cell. It's iterative and it's driven by RecN. What's interesting about this model of search, at least according to us, is that it can span through diverse cellular geometries. Because RecA is going to move from a fixed point to one location and back again, it should be able to sample irregular cell shapes as well. And it doesn't need the entire chromosome to get mobilized. All you need is a filament to be mobile inside the cell. And so the questions now we want to ask is what's the mechanism by which this translocation is driven? And there's two key aspects to this, right? There's the aspect of the chromosome itself. So RecA is moving, but how is it moving inside the cell? Is it moving through the chromosome? Is it moving on the chromosome? And then molecularly, what exactly is RecN doing to RecA that allows it to move inside the cell? Maybe we can postulate an example of treadmilling-like behavior or an example of extrusion of DNA ends that allows for this movement to occur. Am I out of time? I don't know. I've sort of lost track now. I still have time? Okay. So I'm very quickly going to tell you about where we are going with these questions. So one of the questions we're going to ask is how exactly does this break site traverse inside the cell to find the partner? How does it go through the chromosome or on the chromosome? So we want to understand what's the role of chromosome compaction or organization in this process, and what impact does cell volume or cell boundary have on it. And just to get you back to these two cell types, what's really exciting about these two cell types is that you can play around with chromosome density in them. So in both these cell types, you've got 2 chromosomes versus 1 chromosome, but the same cell volume. So the only thing that's changed now is the density of the chromosome itself. Here you have 2 chromosomes versus 1, so there's lesser compaction in this case than here. And so what Saurav in the lab has been doing is been trying to ask, well, what are the rates of movement of RecA? What are the velocities of RecA in these cells versus these cells? And does the amount of chromosome play a role then in determining how quickly RecA can get through during the process of traversal? And he finds something really, really striking. Cells with 2 chromosomes, RecA moves much slower for the same cell volume. So what you see here is the time taken for a traversal from one pole to the opposite cell pole for cells with 2 chromosomes versus 1 chromosome. So this suggests really that cell density is playing a role in determining how quickly RecA can traverse through to find the homologous partner. All right, so I'm going to stop here and remind you again that we've been able to look at RecA dynamics inside the cell, and we are postulating the possibility of a directional movement or traversal to enable the search process. And we are trying to now look at the process and understand what the mechanism is that drives RecA movement inside the cell. I'll stop here, and I'm happy to take more questions.
Thanks, Anjana. Questions, students?
Hey, yeah, thanks, very nice talk. This may be a simple question, but I was just wondering about the comparison you made to E. coli. So you say, okay, these cells have a bit different shape. It's not completely clear to me, are they much, much taller? Or do you think in E. coli it also does this traversal, or is it there just stuck in its place because it's too small or something? How does...
No, thank you for asking that question, because So although the E. coli model is that of a stiff rod in the cell with the chromosome doing search instead, the data are consistent with what we observe. In fact, there is movement of RecA in E. coli as well. And more recently, the ELF lab themselves have also seen that RecN, the deletion of RecN disrupts that movement. So this seems to now hold true, at least across these two bugs, which is very exciting for us.
Thank you, very nice talk. So I was just wondering because about the translocation of the RecA. So there are, I know there are models for other kind of translocation in bacteria that are reaction diffusion models. So I was wondering if you have thought about that.
Yeah, no, thank you. Okay, so I'm going to jump to this slide here. We've considered two models and more recently want to add a third model to the mix. One possibility for RecA itself is the possibility of it treadmilling on the single-stranded DNA, allowing it to move in a directional manner. And so the treadmilling can be driven via regulating the rates at which RecA adds and removes itself from the filament in a directional manner. And in fact, a theory built by Madan sort of supports the idea that treadmilling could explain a lot of the dynamics we observe. That being said, RecN is an SMC, and SMCs are known to loop extrude. And so it's by a motor-driven activity. And given that RecN's ADPase cycle is important for the dynamics we observe, it's also possible that there is some motor-driven action of the break ends that moves it in a directional manner. And finally, more recently, it was proposed by another group that maybe what we're observing is similar to this diffusion ratchet mechanism that's been proposed for some other proteins in bacteria, including PAR-AB, where PAR-B chases after a PAR-A gradient. And it's possible maybe RecA chases after a RecN gradient. We don't have an answer to this one question, which is where is RecN right now? And so that might help us resolve this a little bit.
Okay, thank you.
Any more questions from students before we move on?
Thanks for the talk. I had a very naive question. So the RecN helps the RecA move, but that's a local movement, right? Do we understand the reset mechanism?
Okay, so when you say when it turns, how does it turn? Yeah, no, that's such a good question. So again, I want to show you these very stark movies. So we did this experiment more recently, sort of in the lab, this experiment where he made cells much longer with many chromosomes in them. So now when I say much longer, they're at least 10x longer than the cells I'm showing you here. And just to remind you, in these cells, you see this nice movement, then turning, and then going back. We can make cells like that, which are monsters, and now they have many chromosomes in them. Each of them have the capability of being a template for repair. And so now if you ask, well, how does RecA move here? It's just crazy. I mean, you can see this filament take off. We rarely see these reversals now because we are not going long enough in our imaging. But you also see resolution quite rapidly. It seems to stop at one of the intermediate chromosomes now. So yeah, this reset question and the turn is very, very fascinating, especially because we don't capture it in these monsters anymore. So it could be a cell boundary. It could be, yeah, it could be the boundary that causes the turn, yeah.
You know, just going back to the loopase, but I mean, if it was a loopase, and instead of extruding a loop, it worked in reverse. If the DNA was fixed, then maybe it would just move along the DNA. And is that? I mean, can you just think of a really identical mechanism where instead of forming a loop, it just transverses across along the DNA, and so it would be really the same activity as a loopasing chromosome compaction enzyme.
Just to rephrase what you're saying, then some sort of a zipping mechanism where you bring together the single-stranded DNA along with the chromosome, and then you zip it together. Is that
I mean,
what
imagine
you're...
if you had a loop and you've got two strands of the loop. And suppose that RecA was on one strand and you were feeding the other strand through, then couldn't you think about that as going in reverse and just
okay.
moving along the DNA? It's kind of moving, it's like a gliding mechanism rather than a locomotive, like a motor that instead of it moving along a filament, it's gliding the filament.
Oh, very fascinating. So, I mean, just to picture, maybe I'm not understanding this correctly. Your idea is that RecN might be taking in the chromosome, and so you've got the RecA nucleoprotein filament, and on the other side, you've got this chromosome loop that's passing through RecN, and that might act as a way of allowing for search. Huh. If that's the case, then you don't need a mobile filament, because then the filament forms and it can be stationary, and all you need is the DNA to...
Well, you could do it like that, but if the DNA was fixed, then it might be hard. I mean, I don't know how anchored, it's anchored to the cell wall or something like that. So if the chromosome were somehow fixed, then you couldn't thread the chromosome through a fixed rec on the chromosome.
Yeah, no, it's possible. It's possible. Absolutely possible. Yeah. No, thank you. Yeah, can't rule it out at this point. Yeah.
The RecA seems to be a very important one for the persister cells, right? And so, do you see any logical links between these two? Does the RecA play the double role, or is it necessary for doing this or that? Any links?
Yeah, so we have done these experiments where we've decoupled the role of the SOS response. So RecA has, like you said, a dual role of also regulating an entire DNA damage response. So we've done experiments where we've decoupled the two. So the search process seems to be independent of the SOS response. So they're not coupled together. That being said, it's really, this is like wild speculation. It's very tempting to suggest that movement in this manner can also act like a cargo carrier. RecA can be depositing repair proteins across the chromosome if it's moving in this manner. And RecA is known to interact with many repair proteins. But at least crudely, we can say that the role of RecA in regulating the DNA damage response has no role here. So we can separate the two.
Okay. You mentioned about the search, but how about the fixing of the DNA? It seems that RecA have a look at the DNA downstairs and then going up to the one that is not broken and then come back. But what about the mechanism of fixing the DNA?
Okay. Yeah, so the question, to understand better, the question you're asking is what happens once you have actually arrived at the break site? How do you dissociate? How do you stop movement? How do you stop moving anymore? Is that right? Am I understanding this correctly?
You have a DNA that is broken, right? And then that protein going up to see the another copy that is not broken, and it's got to do something with check something that the broken part is somewhere, and then it come back to the broken DNA. But what happened after that? Does it fix the broken bit? And that protein must have some kind of memory that remember the number, the location of the broken DNA. Suppose base pair number 100, and then it's got to fix that particular location.
So there's a recent structure that sort of helped give some insight into this. So there's a two-step mechanism by which RecA does microhomology sampling. So every time RecA interacts with the double-stranded DNA, it's going to do local base melting and try to do base pairing. And so it does this in two steps. So one is a 5-base pairing. So if RecA which is carrying single-stranded DNA, that single-stranded DNA is able to form a triplex, so it ends up base pairing efficiently with 5 bases. Then the rest of the filament invades and stably does base pairing, and then the RecA filament dissociates completely from that break site. And that's when you start to get repair. But during the process of microhomology sampling, when it's not successfully being able to pair, it'll reverse and come back again. So 5 bases is what gives the initial step to license RecA to dissociate completely. So it doesn't happen in the first go. So many times what we think is happening is that RecA is microhomology sampling, trying to make this 5-base match, and if it doesn't, it lets go and then keeps moving. And that's why we see it turn around and come back also. Now, once it actually finds the break site, our guess is that what ends up happening is that the stable base pairing happens and then RecA dissociates. But what really is the molecular detail of it, we don't know as yet. That being said, I don't know if RecA has a memory of exact base number, so I don't think it measures where the break is and how many bases away from the break site is. We say this because in a cell with just one chromosome also, you get movement, which suggests that the movement and search is an inherent property of the filament and nothing to do with the presence of a homologous template. So is there a difference, though, between the chromosome where the break occurred and the chromosome where no break has occurred? That's an interesting question. It's possible there's something different about the topologies of these two chromosomes that allows for RecA to sample them differently. And we are trying to explore that possibility now.
All
Thank
right. Let's thank Anjana once again for a wonderful
you.
talk. Our next speaker is Pablo Sartori, who joins us from IGC.